we're very interested in hearing more from him today. So prior to coming here in 2020, which must feel like just yesterday, given the effects of the pandemic, Fei Zhao began his career, got his bachelor's degree in China at the China University of Mining and Technology, focused on bioengineering. And then after that, shifted to a PhD in toxicology at the University of Georgia. Right before coming here, Fei Zhao did a postdoc at the National Institute of Health, at the National Institute of Environmental Health Sciences, where it really crystallized his interest in developmental biology of reproductive tract. And that's what we'll hear about today. He came here with a K99 ROO award to continue to study this subject. And the first 2 years of his lab here, despite the pandemic, have been very productive. And I think, I want to note that one wonderful thing that I can clearly see from both your application to be a trainer and your website is your deep commitment to mentoring. So I just want to say congratulations on taking all of your students and lab members to the meeting this summer and on the awards they got there. And these are some of the reasons we think Fei Zhao is just going to be a fantastic genetics trainer too. So hopefully you'll agree. Without further ado.
Thank you so much, Nico. It's not easy to memorize all the institute that I attend, and thank you for taking effort on that. Hello, everyone. Are you sure you guys don't want to sit in front row? We got a very small screen today. All right, hello, everyone. So I would first of all thank this wonderful opportunity by the genetics program for me to present and share our research with you today. I really want to hear some feedback and comments from all of you. And today I'm going to talk about a discovery that we made, and then discuss how this discovery leads to a few ongoing projects that is focused on my current lab. So my lab is interested in how an embryo establishes such a dimorphic reproductive tract system. So this probably organ, maybe when you dissection, you throw away, but this is very critical for our reproduction. So without here, I will not be here. So without this organ of my parents, I will not be here today. And in the female reproductive tract, in mouse model that we are using and other mammalian species, the female reproductive tract includes ovula, uterus, cervix, vagina. These organs are critical for pre-implantation, embryo transport, and development, implantation, presentation, and the support pregnancy, as well as provides a critical passage for delivery during maturation. So on the other hand, the male reproductive tract include the epidermis, vas deferens, femoral vesicle, and these organs are critical for sperm transport and maturation after it's released from the testes. So although this system displayed cytodimorphism, so at the early beginning, so before sexual differentiation, regardless of sexes, an embryo possesses identical reproductive system. So both male and female possess paired undifferentiated donor and the two sets of reproductive tracts. So Wolfen duct, this is progenitor for the male reproductive tract, and the Muller duct, and this is progenitor for female reproductive tract. So during sexual differentiation, the gonad will first differentiate into the testes, which will produce two hormones, androgens, which will promote the differentiation and the maintenance of the Wolfen duct. And the second hormone is anti-Muller hormone, AMH, which will induce the regression of the Muller duct. So consequently, in the male embryo, so which tract will be maintained? So which tract will be maintained after the action of two hormones? I'm sorry? The Wolfen duct, thank you. And the Wolfen duct will maintain, so the Muller duct regresses, so consequently, the male tract the Wolfen duct will eventually give rise to the entire male reproductive tract organs. Conversely, in the female, the gonad will differentiate into the ovary, which does not produces two hormones. As a result, the wolfen duct degenerates and the maroon duct is maintained and eventually differentiated into the entire female reproductive tract organs. So this system, the fate of the two reproductive tracts, it depends on testicular hormone action, which will drive the male phenotype. So the female phenotype is considered default because it will occur and only occur in the absence of testicular hormone actions. So if you make a cross-section of the fetal tissue, you will immediately see the two primitive ducts, the wolfen duct and the maroon duct, are surrounded by their own mesenchyme, the wolfen duct mesenchyme and the maroon duct mesenchyme. So mesenchyme is connective tissue. It's undifferentiated connective tissue that dictates the epithelial cell fate and differentiation. So the androgen works on this receptor known as androgen receptor to promote the maintenance and differentiation of wolfen duct. And anti-neural hormone AMH works on this receptor, anti-neural hormone receptor type 2, for inducing maroon duct regression. So it's well established the mesenchymal signaling dictate the fate and differentiation of ductal epithelia. We are very interested in the mesenchyme and looking for novel mesenchymal factors that may play a role in reproductive tract development. And we identify a transcript factor known as COOP-TF2. It's specifically expressed in the mesenchyme surrounding the wolfen duct and the maroon duct in both male and female embryo. Okay, given the significance of the mesenchymal cell in reproductive tract development, the specific expression of COOP-TF2 in the mesenchyme suggests its potential role in reproductive tract development. So we decided to study the functional significance of this transcription factor. One thing I forgot to mention is COOP-TF2 is an alpha-nuclear receptor. Has been implicated in organogenesis of multiple organs. And however, the COOP-TF2 embryonic COOP-TF2 global lookout leads to embryonic lethality. So we decided to use tissue-specific conditional knockout mouse model, where we specifically delete COOP-TF2 in the mesenchyme. So we gathered the mice and then perform hormonal immunostaining of epithelial marker PAX-A to visualize the presence of the reproductive tract. So here in the control, you can see one single reproductive tract. This is the maroon duct. Here is the ovary. So what will happen if we delete the COOP-TF2 in the mesenchyme? So in knockout female, we found there's two reproductive tracts, both maroon duct or wolfen duct is maintained. And we also perform wolfen duct marker to validate the identity of this ectopic maintained duct. This is definitely wolfen duct. Okay, so, and one thing I want to point out, if you look at the shape of the ovary, the control is heart shape. It's like very beautiful heart. This one is very elongated. This is due to the physical presence of the ectopic maintained wolfen duct. So remember what what control the fate of the wolfen duct? Which hormone? Androgen, right? So androgen controls the fate of wolfen duct. So based on the current paradigm, so the testis-derived androgen works on receptor to promote wolfen duct maintenance. In the ovary, in a female, the ovary does not produce the hormone. As a result, wolfen duct degenerates. So the ectopic maintenance of wolfen duct in a female may suggest there's ectopic action of androgen. So is there any ectopic action of androgens? So to address this question, we perform two studies. First, we get to the knockout ovary and to examine the expression of some key enzyme for androgen production. So HUSD3 beta and the CYP17A1. So these are two enzymes for androgen production. So in the testis, we have abundant expression of this stereogenic enzyme expression, but in the white type and knockout ovary, the expression is minimum. And more importantly, the expression is comparable between white type and knockout ovary. Another experiment I did is I collect methanophores and then to check the expression of androgen receptor and the androgen responsive gene. So androgen receptor expression is comparable between white type and knockout female and as well as the control male. And this two androgen responsive gene is highly expressed in the male where there is androgen action, but it's very low in white type knockout female. So these results suggested that maybe androgen does not play any role in the Wolfenbach maintenance in knockout female. However, I was not convinced because it's, it had been, the maintenance of Wolfenbach had been exclusively linked with androgen action. So I want to prove conclusively that there's no androgen action in knockout female. So to do that, I was inject the dam that carrying knockout female embryo with androgen antagonist, androgen receptor antagonist, flutamide, during the window, during masculinization programming window, and then collect the samples at birth to see whether the Wolfenbach will maintain or not. So I want to point out this is well-established treatment that can block the androgen action. So in the oil treatment, in the knockout tissue, there's two reproductive tracts. So the Wolfenbach will be is maintained. I argue if androgen play a role, this treatment will block the maintenance of Wolfenbach. So we will now see Wolfenbach maintenance. However, it was not the case. Even though I block androgen action, the Wolfenbach is still there in knockout female. So those results suggested that the Wolfenbach maintenance in knockout female is not due to androgen action. So what could be the mechanism? So I was very puzzled at the time, but very fortunately surrounded by very talented and creative lab people and the neighbors lab who are working on nuclear receptor, epigenetics, stem cell. They give me a lot of wild ideas. I test a bunch of them, and then eventually I pay attention to some locally produced growth factor, FGF, EGF. This is two. So this is two mesenchyme-derived growth factor that have been involved in Wolfenbach development. So these two growth factor both working on its receptor specifically expressed in epithelial cell to control Wolfenbach differentiation. I examined the FGF and I examined the EGF and the EGFR expression, and there's no change. However, when you examine the FGF and FGF receptor, I found some differences. So here is a qPCR of the expression of the major component of FGF signaling pathway in the white type and knockout female methanophores. Here is the major receptors in the tract, in the Wolfenbach, and here is the major ligand for these receptors. So we observe ectopical, we observed the enhanced expression of FGF and FG7 and 10 in knockout female. So this result suggested that CoopTF2 ablation increases expression of FGF 10 and 7 and 10, which potentially can involve in the maintenance of Wolfenbach. Then we asked, could we add the FGF7 and 10 to reproduce the Wolfenbach maintenance phenotype in a white type female? So to address this question, we collect those samples, control female urinary genital range, and culture for 28 hours and perform hormone tax 2 staining. So in a vehicle treated group, after culture, the Wolfenbach successfully degenerated. In the presence of FGF7, 10, and or their combination, we observed ectopic maintenance of the Wolfenbach. Okay, so these results suggested that activation of FGF signaling may reproduce CoopTF2 knockout phenotype in the white type female. Then we asked if there's ectopic FGF signaling, and we usually see the activation of intracellular signaling components. So once FGF binds to its receptor, it will activate and phosphorylated two component AKT and MAPK, also known as ERK. So we perform the immunohistochemistry to see which pathway is actually ectopically activated. So in the control group, we see the PERK staining is very low. However, when we see examine PERK suppression in the knockout female, we see ectopic maintenance, ectopic enhancement of PERK staining in the maintained wolfing duct in knockout female. So this is the result suggests that ERK pathway is a key downstream mediator of enhanced FGF signaling in knockout female. Then we ask if it is true, and if we block ERK pathway, we can potentially rescue the phenotype. So to address this question, so we culture the knockout female. So in knockout female, without any treatment, the wolfing duct will maintained, just like in a in vivo situation. In the presence of PERK specific inhibitor, the wolfing duct degenerates, and we only see the Müller duct is maintained. So these results suggested that ERK activation is responsible for maintaining the wolfing duct in knockout female. So far, I've been talking about knockout female. What would happen if we remove CoopTF2 for the wolfing duct? So what does this mean for the normal condition? So in the knockout female, when we remove CoopTF2, there's enhanced FGF production, which will activate this receptor expression amphitheater to turn on intracellular PERK-mediated survival pathway. So what does this result tell us? The function of CoopTF2 in normal condition. So in normal condition, the CoopTF action is to actually suppress FGF to safeguard the programming, the regression program. So this work actually is contrary to our current paradigm. Looks like the wolfing duct maintenance, wolfing duct regression is not by default pathway. It's not default outcome. It's not passive outcome due to lack of androgen action. It is actually actively promoted by CoopTF2 in the mesenchyme. So this work was published when I was a postdoc in Atlanta EHS in North Carolina. Then when I moved to UW-Madison, we remained the same interest in the wolfing duct mesenchyme, but ask a different question. So I hope you still remember there's two ducts at the beginning. Okay, they're surrounded by their own mesenchyme, the wolfing duct mesenchyme, the Müller duct mesenchyme. So the wolfing duct mesenchyme is specifically expressed GLI1. This is readout for this mesenchyme, the fetal stage. And anti-Müller hormone mesenchyme, Müller duct mesenchyme specifically express MHR2. So these are two different mesenchyme. They not only express different gene, but display some different cellular properties. So during sexual differentiation in the female embryo, the wolfing duct degenerates. And then what happened to the surrounding mesenchyme? Are they eliminated or become part of the female reproductive tract? So given the significance of the mesenchyme in organogenesis, we are very interested in the question and decided to track the fate of the GLI1 positive wolfing duct mesenchyme. To achieve this goal, we performed a genetic lineage tracing model. For some of you who don't know, I apologize, I don't have the background slide, but what happened is in this genetic lineage tracing model, GLI1 Korea rosa tomato, you can permanently label some of the GLI1 positive cell with red fluorescent protein so that you can trace the fate and those cells and their progeny as they differentiate. So this what happened in this technique is that I successfully label some of the mesenchyme surrounding the wolfing duct, and then before sexual differentiation, so some of the cell surrounding the wolfing duct actually labeled with red fluorescent protein TD tomato. And then 2 days later, when the wolfing duct degenerating, or completely degenerating on 18.5, those labeled Wolfendam mesenchyme are actually still there. So that means the Wolfendam mesenchyme is not eliminated with the epithelial cell. They are maintained and become part of the female reproductive tract. So one thing I want to mention is that all my picture here is oriented. This upper part is the mesometrial side. This part is the anti-mesometrial side. So what happens is this is the tract. They have one side connected to the systemic circulation and body cavity. So this is the side, mesometrial side, where you will see a lot of blood vessels. So this is a lot of important for the reproduction because in mice, placentation is only occur here, and implantation only occur on this side. Anyway, so it looks like this cell, the Wolfendam mesenchyme derived cell, are predominantly localized at the mesometrial side. So they have, and then the next question, what about the Wolfendam mesenchyme derived cells? So similarly, we use another lineage tracing, but this is slightly different. It is Cray, Rosa tomato. This basically label every cell that express MHR2. And what we can see, and those Wolfendam mesenchyme derived cells exclusively localized at the anti-mesometrial side, but never found in the mesometrial side. So this result suggested that Wolfendam mesenchyme contribute to an anatopically unique mesenchyme population in the female reproductive tract. Then we asked another question. So there's two populations for forming the female reproductive tract. So how these two mesenchyme populations different? To address this question, we use cell sorting, get the cell out, and compare their transcriptomes. I'm not going to show you the 1,500 differential expressed gene and tens of gene signaling pathway, but I'm going to show you the marker we identified for the two populations. So one marker for the Wolfendam mesenchyme derived cell, remember this more localized at the mesometrial side, AKP12. This is a kinase protein, kinase acrin protein that plays a critical role in cytoskeleton and the cell migration. So this expression at birth and at early stage is enriched at the mesometrial side. On the other hand, the HAM2 is a transcription factor specifically expressed in the mesenchyme at the anti-mesometrial side throughout the developmental stage from 14.5 through the 18.5. So with these results, I'm very confident to say that the female reproductive tract, for forming female reproductive tract, there's a dual fetal origin of mesenchymal cell. And also this Wolfendam mesenchyme contribute to not only anatopically distinct, but also transcriptionally unique mesenchymal population. So what I've been talking about so far is just characterizing of the phenotype of this cell population. So what's the functional significance of the Wolfendam mesenchyme in the female reproductive tract development? To address this question, I'm using the genetic cellular ablation model. So in this model, it's quite similar to the genetic lineage tracing, but instead of induce fluorescent protein expression, you induce a toxin expression known as DTA. So you can induce the DTA expression specifically in GLI1 positive Wolfendam mesenchyme to see what would happen. So one thing I want to point out that this DTA, this toxin, internalization of this toxin require a specific receptor, which is not expressed in mice. So once they kill those cells, the toxin released from the environment to the environment, they will not influence the survival of the surrounding cells. So it's very specific and targeted ablation of this population. And the GLI1 actually expressed in multiple tissues, and in vivo injection of tamoxifen to inducing cellular ablation will be problematic because that will lead to embryonic lethality. So to circumvent that potential problem, we're using ex vivo organ culture. So in this experiment, we collect those samples and from control and ablation group. At this time, we do not inject tamoxifen, so there's no ablation at the beginning of the experiment. So after putting culture with tamoxifen and inducing the cell ablation, so in the control group, I hope you can see there's some expansion of the maroon ducts. There's some coiling at the cranial part. This is where we're going to form very coiled orbita and expansion of the lumen. However, in the ablation group, the expansion and growth of the maroon duct is standard. We do not see significant coiling at the oviduct region and significantly reduced lumen expansion. So we of course cut sections to make sure this reduced growth is due to the lack of epithelial cell. And we also do some work to identify some growth factor that's secret from the Wolfenbach mesenchyme. So those results indicate that the Wolfenbach mesenchyme not only contribute to become part of female reproductive tract, but also play a significant role, potentially significant role in female reproductive tract development at fetal stage. So far, I've been talking about the female. I want to little balance here. So the female, as I mentioned, the Wolfenbach, in the absence of androgen, they are going to, the Wolfenbach degenerates and those mesenchyme are going to become part of the female reproductive tract. It can be, this fate can be designated as a female fate. But we know this mesenchyme, Wolfenbach mesenchyme, have the bipotential. If you give the female some ectopic estrogen, androgens, you can masculinize this tissue and make promote their maintenance and differentiation into the male reproductive tract, just like its counterpart in the male embryo. So next we're going to talk to fully understand the sexual differentiation of Wolfenbach mesenchyme. We are curious about androgen action and ask how does the androgen regulate the Wolfenbach differentiation and promote the male fate of Wolfenbach mesenchyme. So the androgen, as I mentioned, is the action is mediated by a nuclear receptor known as AR. So when the androgen here, the example one of androgen, testosterone, once bound to the receptor, they're going to form the dimer, move to the nuclei to bind to this regulatory region to control AR target gene expression. So AR is very important and is expressed in both epithelial and the mesenchyme. As I mentioned, mesenchyme critical role. So does the androgen receptor in the mesenchyme is very essential. So this concept that the mesenchyme is critical for tissue for ductal morphogenesis and differentiation is based on tissue recombinant studies, but no one had done in vivo to demonstrate the developmental requirement of androgen receptor for Wolfenbach differentiation in the male. So in the presence of androgen action, and the Wolfenbach will first stabilize in the male, and then undergoes extensive coiling from E16.5. So to address the significance of the androgen receptor in the mesenchyme, we are using another cray that specific target the mesenchyme cells surrounding the Wolfenbach. This is OSR2 cray. It's a very nice cray that very efficiently can ablate the cell in the mesenchyme at early stage. So in the control, and I hope you can appreciate there's a coiling of the epidermis. This is actually this is due to elongation and preparation of the feeder cell. This coiling is very essential for male fertility. So if you can straighten the epidermis, human epidermis is about 3 or 6 meters long. So why it needed this long and extensive coiling? Because the sperm need to travel long enough to get sufficient nutrition and developmental factors for their maturation. So what will happen if there's androgen receptors absent in the mesenchyme? So in the absence of androgen receptor in the mesenchyme, the Wolfenbach lack its castrate coating and remain a straight tube. And we even see some cystic formation. So this is something we are currently working on, and we are trying to using histology and immunohistochemistry and some genomic approach to understand what will happen and the underlying mechanism in this phenotype. So another thing I want to talk about is something related to androgen receptor. It's something very I've been pondering over for several years, is that androgen receptor in band-specific space. However, the content in most cases is inaccessible. So the DNA wrap around the histone forming histone around the histone forming the nucleosome, and the topological organization of histone and nucleosome will actually block and make it not perceivable for androgen receptor action. So in order for masculinize the Wolfram duct, so the comping region that for androgen receptor must be accessible, so that AR can bind this region to turn on developmental gene expression. So to address how is the comping accessibility landscape is established for androgen receptor in the mesenchyme during this critical window. And we are get the sort this Wolfram duct mesenchyme from the male where there is androgen action, and from the female where there's a lack of an AR and action due to the absence of androgen ligand. We collect those samples from 14.5 and 16.5 from male and female. This is window of sexual differentiation of reproductive tracts. And we do the profiling of those cells and the assay their comping accessibility. So we found that there's during this sexual differentiation, we only found around 1,700 unique accessible accessible region in the male. So here is a heat map of showing the accessible region signaling in this unique masculinization associated accessible region. So in the female, there's no opening, but in the male, over the time, seems the region is gradually opened. So when we perform motif analysis to understand what factors that potentially enriched in this region, we found actually androgen receptor and its motif, ARE, and half size enriched. So these results suggested that those chromatin accessible region might mediate androgen receptor transcription program, and this region actually could be opened by androgen receptor. So I only talk about a small portion of chromatin accessible region. It's about 17,000. This is only about less than 2% of all the chromatin accessible region. So except this small portion, the older groups, the chromatin landscape is almost identical. So they are same. It makes sense because there's two reasons. One, they're still the same cell type. They're still mesenchyme. They are not changing their huge fate. There's no huge fate change. Another thing is that, remember, the female can be masculinized by androgen action as well. So the female also get ready. And so the chromatin accessible landscape is quite similar between the male and female. Now I'm wondering what factor control the accessibility of this region? And in the field, there's some theory about known as pioneer factor. So those factor is a class of transcription factors that have the capability to bind to condensed chromatin and open it for facilitate androgen receptor transcription program. So in the field, based on my knowledge, so the pioneer factor concept is first derived from cancer biology, and then there is several pioneer factors that known that essential for opening chromatin for androgen receptor. There's GAT2, GAT1, 2, 3, 4, 5, 6, and the HOXB17 and FOXA1 and FOXA2. And we examine those known pairing factor and their expression in the mesenchyme. Only one actually specific expressed in the Wolfenbach mesenchyme. And this one is GAT2. GAT2 is specifically expressed in the mesenchyme surrounding the Wolfenbach duct. Because this is the only pairing factor that expressed in the Wolfenbach mesenchyme, and we wonder whether this is GAT2 is very critical for Wolfenbach development. So my argument is that if GAT2 is critical for androgen receptor transcription program, when we knock out GAT2, and we should see some defect in Wolfenbach development. Is that the case? So to address the question, so we cross this flux mice with the crate we are using to knock out the AR to see what happens. So in the control group, again, in the epidermis, there's very coiled, extensive coiled epidermis here, and this is essential for the men's fertility. Before I show you the result, I had to let you know our lab knocking out 5 genes, no phenotype. Do you know how frustrated I am? And I still don't know whether there's any phenotype. There are so many critical transcription factors and genes that play many roles in kidney, in the lung, in the heart. I thought will be the same factor will be critical in the reproductive tract. No. So we are very anxious and to see whether there is knockout when we knock out GAT2. So it turned out it is. So the Wolfenbach lack of this characteristic coiling and remnant street 2. I think there's still a lot of things to do, but this functional result give me the confidence that maybe GAT2 has some interaction with androgen receptor. One of them could be a GAT2 player pairing factor. So this is another project we're working on. As I tell you, this is ongoing projects, and I would like to hear some of your feedback and the suggestions. Okay, I think I'm going to summarize what I've been talking about today. So I focus on one population, Wolfenbach mesenchyme, and talk about its 2 fate. So the unexpected role of the Wolfenbach mesenchyme in the female and new perspective on androgen action in the male. So in the female, as mentioned, the Wolfenbach degenerates. This is not default process. It's actually actively promoted by Colloquium. Once Wolfenbach degenerates, those mesenchyme remain and become part of the female reproductive tract. So there's a dual origin of mesenchyme population for forming female reproductive tract. On the other hand, I also talk about the male, where androgen will driving the Wolfenbach development. And I will demonstrate the essential role of androgen receptor in the mesenchyme, and also providing accessibility of the Wolfenbach mesenchyme, and suggested that AR and GAT2 may interplay in promoting Wolfenbach differentiation and establishing coupling accessibility. With that, I want to one thing I need to mention, because I was keep being asked, is that okay, the Wolfenbach mesenchyme become a part of female tract. So what about the Muruda mesenchyme? Are they become part of the male? Yes, we have the preliminary data. I don't have time to show, but yes, there are some of them, not many, actually still maintain and become of the male reproductive tract. So with that, I would like to thank my lab people. I'm very new lab, but I'm very fortunate to recruit great people, Shuai, Jillian, McKenna, and 2 undergraduates. My first PhD student just joined the lab. You know how thrilled I am. So I feel very privileged to have the opportunity to work with my students. With that, I'd like to stop here and let's end questions.
I was just wondering, did you look at what controls the expression of QPS and what's the difference between males and females, and how it interacts with androgen?
Yeah, that's actually a great question. We are also curious about what control QPS expression. In the field, there's 2 hypotheses. One is studying a tract player role, And another thing pathway is red amino acid. We tested the hypothesis. We knocked out the chop. We blocked the red amino acids using pharmacological approach. Cooked research not changed. So we still don't know what happened. So what controls this expression of cooked mesenchyme? Another question is the potential interaction of cooked with androgen receptor. We do. At the beginning, we thought they work together or antagonize each other because androgen receptor promote the hormonal differentiation, cooked to suppress its maintenance. So, but we performing the chip-seq and analysis found their binding have very few overlap. So we now thinking they may be activate or control that rate pathway to control the open fate.
And are the QTS actively repressed in males?
So in the males, androgen receptor actually quite dominant. What happened is I think they suppress one program, but androgen receptor turn on the survival pathway and the overcome it.
So you're saying the QTS expressed equally?
Yes, in both male.
I have you done some kind of chip-seq or something to see if QTS is actually able to bind to the target size in both sexes, or maybe just
Yeah, we control the binding site as similar between the female and male. And androgen receptor binding and QTS binding is very different. They have very few overlap. So I know it's unexpected because QTS2 and androgen receptor both belong to the same steroid.
I was asking if the QTS bind to both the female and male.
Yes.
The same size.
Same size.
Not compared to multiple. I'm just the QTS in male versus
Same size.
so they have it.
They have same thing.
Mhm.
Yeah, I know.
But that's very interesting.
Yeah, I hope they're different. I had to look. I had to test my data.
Yeah, it takes more than one design for you.
I'm curious, do you know how evolutionarily conserved some of these regulatory pathways might be? Like, is there wolfing duct mesenchyme associated with development in teleost and cartilaginous fishes? Like, how much do you know about the evolutionary breadth of this?
Yeah, good question. So not that much. So the QTF were first identified in chicken. So actually, is I would say QTF is expressed in the bird. But beyond that, I probably ask the I'm sorry.
Fruit flies have homology.
Oh, right.
We
Okay.
actually studied it in the lab.
Okay. Okay. They're very Chinese more about that. Okay. Yeah, it looks conserved, but but I know yeah, in the bird, in the chicken, the species is there. Even in human case, we have not how there's some sexual reversal, but it's more some defect in the gonad stuff the tract. Looks like although you convert the conserve, but there's a species specific function.
So another follow-up question, which probably puts it off topic now, but there are a number of vertebrates which are sequential hermaphrodites. And so they will like digest and redevelop reproductive tracts in various ways. And I'm curious if you think that would be a useful model system for like would you think that would be a similar developmental switch, or you would be like a different developmental program, or it's a little off topic.
Okay, invertebrate. You have to repeat the question. You say you are the
So like there are wrasse or clownfish or like a number of species. There are a number of species that have very different sex chromosome systems. So there's not like an XXXY.
Okay.
So the genetic determinants or environmental determinants that they
Right,
have
right, right.
genetic determination system are very different. And they can also be sequential hermaphrodites. So they can have their development of their primary sex organs is not terminal. They reach one developmental state and then they will do a subsequent development. And I was wondering if you had any thoughts about how that might differ from this initial development in a genetically determined XY system without sequential parameters.
Yeah, that's a great question. I think they have different mechanisms. You are talking about the temperature dependent or environment dependent sex determination system. Is that
That's part of the question.
yeah, I think I'm not aware about that. And in mammalian species, there definitely is XRY. But I know, especially the fish, oh my God, every species has a different mechanism. And I think even they can change the fate based on the social cue and the presence of male or female. That's really fascinating, but I'm not sure about how comparison of those. But definitely I will keep an eye on that.
They might converge to something different.
Could be, I think so.
We should also check and see if there are any questions from the online group. Sorry, I'm not connected anymore. I would check there. Chat. That would typically be in chat. Okay, I don't see at least a.
Oh, I don't think I have questions here.
Okay, then we can take another one here.
Okay.
You tend to test the influence of external factors on the fate.
Yeah.
Cell, because you have a bit of mechanism in cell biology.
Yeah. Yeah.
And which one?
You mean environmental factor?
Yeah.
Yeah.
You showed that oncogene
Yeah.
is not involved in the tomato fate, for example, but other things can probably affect the fate of these cells, right?
Yes.
I don't know, some diet, some very new, like non-biological factors. So
Yeah, that's a
do
great
you
question.
intend to test?
Yes, I'm intent to, and we more than happy to, really want to. So, yes, because for major reason, the deep birth defect is combination of genetic and environmental factors. So it's definitely environment. We cannot overlook the environmental factors. Actually, I was in the toxicology background. I working on estrogen and endocrine disruptors. So, my plan is once I get my first R1 of this part, then I can explore the environment. If you want to see one environment factor that I'm very interested in, I still want to working on the estrogen, synthetic estrogen, yes, because it's a toxic endocrine disruptors, and the drug is prescribed to pregnant women in 1960s and caused millions of women offspring as a defect. And that's something I'm still working on, what we really want to do. The major reason, another reason is DS treatment at fetal stage actually can formalize the male reproductive. So if you give DS to the male tract, and the seminal vesicle part will display the uterus-like structure. So that's something I'm very interested in. So although it's probably no one uses DS, but it's still a classic model to study how environment influence the reproductive tract.
Maybe I understood when you're talking about the like is that of the content fertility, and you said there's like a potential like a list of like factors that could be like say off this something. I was just wondering like I meant this is the do you have like a motif and like do they share like a common motif like that how you see a change in the like you got to motif openness.
Yeah, so in that complex accessible region, there's hundreds of motifs. GAP2 motif is a small portion. So there's two possible motifs. One is GAP2 may not play a role, or they play critical role. They just do not need to modulate all the region, but they only establishing the region that very critical for the receptor action. So I think we're still working on that.
Yeah, really interesting talk. I'm wondering if the transition to male or female, if that's you having a communication that's like an amplifying move. So when you keep going, it's amplified and several people like a walking mode. And related to that, it's also bilateral. There's two sides. And if you have a compromised situation, you might have the transition like this. So asymmetry, I wonder if you see that asymmetry in some intersections.
Yeah, it's a great question. So I address the second question. We compare the two, but in some cases, especially human, only one side factor. And symmetry is we observe. I do not pay attention to that when we are randomly collect side and do the section. I would imagine because I random select, which would be both sides. But we do see, and there's a report in the literature, and we mark how gene, it looks like one side more likely to affect it. So that's something we are very pay attention to, the symmetry of the phenotype and potentially asymmetry of the gene program between the left and right. And your first question you are talking about quick decision, can they stop? Is that something you're talking about?
Like whether they get amplified, once you get going from that way, it's small. That's the application cascade.
So you are saying that during quick decision, there's a signal pathway to control the fate.
Like an amplification.
Like a positive feedback.
Positive feedback.
Yeah.
That's a good question.
We don't really show what the asymmetry also. So like, if the other intersects and both of them have like this, but it's that amplification, we might have two sides.
Yeah, good question. So I think that's totally possible. I think I'm not very familiar with the genetic loop, and I'm treating the field of recovery, but definitely want to more. Thank you.
Okay, if you have no more questions, let's thank Fei Zhao again for the third.